Hi friends, welcome to this video, and this video will discuss the organization of the eukaryotic chromosome. So chromosomes are found in the nucleus and these are linear double-stranded and are fixed in number. So morphology, the heterochromatin, the nucleosome, eukaryotic, and the metacentric, submetacentric, ectocentric and telocentric are the positions of the centromere in the chromosomes. So nucleosome as a functional particle. So eukaryotic DNA is tightly bound to a repeating array of DNA histone protein particle called as the nucleosome, and which is the unit of the chromosome. So proteins contain the histone and the non-histone protein. Histone is a positively charged basic protein, contains 5 classes, that is H1, H2A, H2B, H3 and H4. And the non-histone proteins are the negatively charged acidic proteins. So nucleosome contains two copies of the four nucleosome stones and forming the histone octamer. So stereospecific bond and nucleoplasmin protein that is negatively charged stable the nucleosome. So H1H4 histone protein binds to a linker or a spacer DNA, that is H1 protein. So histone octamer, and here's the beaded structure of the nucleosome. So 30 nanometer chromatin fiber. 30 nanometer fiber has a two model, that is solenoid model and the zigzag model. Top view of the 30 nanometer is solenoid model. Linker DNA doesn't pass through the central axis, and in zigzag model the linker DNA passes through the central axis. So high order of the chromatin structure, the the proteinaceous structure known as a nuclear scaffold, compact the DNA structure. So an average molecule chromosome could contain approx 2000 loops to mean. The two classes of protein that contribute to the nuclear scaffold having identified one of these topoisomerase 2 in the protein scaffold. This figure showing the loops of the chromosome, 30 nanometer in the length, and the loops, there are so many loops, 300 nanometer in this width area, and non-histone scaffold. So nucleosome assembled immediately after the DNA is replicated. So level of packaging in the chromosome is described as nucleosome with 11 nanometer, then the formation of the solenoid, that is 30 nanometer, then a super solenoid which is of 300 nanometer, and then chromatid formation, and then 1400 nanometer which forms the chromosome. So the figure describes much better. The short region of 1 nanometer is equal to 100 angstrom. There is a 0.2 meter in nanometer the width, and then 11 nanometer, then 30 nanometer form the solenoid like structure. The loops are formed of 300 nanometer, and ultimately it formed the chromosome. And this is the area where this describes 700 nanometer densely section of the metaphase chromosome. So all about this. Thank you, keep watching.